Our discussion on Chapter 24, Digestive System Infections. And these are the learning objectives. So for this chapter, it's kind of a long chapter in the book, and it talks about a lot of different topics. I'm only going to focus on gut microbes and the bacteria part of this chapter. So learning objectives, be able to describe how our digestive system protects us from pathogens, ways our gut microbes can benefit us. We'll talk about food intoxications, some different intestinal infections. We'll relate those to climate change, and finally a common healthcare associated infection that some of you may deal with, Clostridium difficile. Okay, overview of the digestive system. This book spends a lot of time going over the different parts. I'm assuming that you already know most of these, so I'm not going to spend time on that. But I do want to talk about how the system protects us from microbes. So kind of starting at the top and working our way down. First we have our mouth. We have the movement of fluids in our mouth. We also have something called the lysozyme in saliva. You might remember this from back in our discussion about the innate immune system, but that damages bacterial cell walls and can help damage pathogens. We also have a lot of normal microbiome in our mouth and throat that compete with pathogens. As the food moves down, most things don't get through stomach acid very well. Very, very acidic, very low pH, somewhere around pH 2, 1.5 to 3.5 is a normal range. That kills a lot of things. After the stomach, once it gets into our intestines, there's a lot of microbes there. They compete with pathogens, and they also produce their own sets of antimicrobial chemicals. Some species of bacillus are really common in our guts, and they produce the antibiotic bacitracin. So we have natural supplies of bacitracin killing off pathogens in our intestines. We also have a special type of cell called M cells. These are important. They're like spies. They find pathogens in the intestines, and they grab them and pass those antigens directly to white blood cells. So inside these M cells, running right next to them, there are capillaries just full of white blood cells. There's little pockets of white blood cells just sitting here in the M cells. And the M cells are constantly spying on whatever pathogens are going by and handing those antigens directly to the immune system to find and kill them. And everything in here, of course, is coated with mucous membranes, those dense tough layers of cells that protect things underneath and produce mucus. And there's constant movement of the muscles in this system and fluids and that mucus that pushes a lot of pathogens through. So I did want to talk more about the gut microbes. They are a major part of our lower digestive system in particular. Once our fully formed feces or waste comes out, it's about 25% microbes in there. So there's a lot of microbes involved in that system. We mentioned they protect us from pathogens, but they also work with a lot of other functions in our body. They aid in digestion. For example, species in the group Bacteroides, they digest complex carbohydrates. Otherwise, we would not be able to digest these at all and get the energy and nutrients from them. There's also a bunch that produce nutrients. There's a group called lactic acid bacteria. The products of their metabolism include vitamins B12, folate, and riboflavin, and we absorb that from the leftovers from our gut bacteria. E. coli produces our vitamin K. This is most of the vitamin K that we get. There's not very much in our diet, and we absorb that from E. coli. There's also a lot of recent research that these microbes interact with our metabolism and our immune system. There have been quite a few studies recently linking gut microbes to rates of type 2 diabetes, allergies, fat metabolism, even our white blood cell production, and among other things. So next, let's talk about some of these bacterial diseases. Some general things to understand. So when we talk about like foodborne illness or food poisoning, there's two basic categories that these are in. First, as illnesses caused by growth of the actual microbes colonizing the GI tract or our intestines. And these operate a little bit more slowly. Symptoms start in a couple of days because that's the time it takes for the microbes to be growing and multiplying and starting to damage our cells. The other type of foodborne illness is presence of a microbial toxin. when you're intaking a lot of toxin, even if the microbes themselves are dead or killed off quickly, that dose of toxin acts very quickly. And these symptoms start within an hour or two of eating that. So very fast onset from those. Another thing to know is by looking at the symptoms, they're all really similar. So to get an exact diagnosis, you have to do more specific testing for which actual species of bacteria it is. Most of these resolve themselves within a couple of days. Most cases, the body gets rid of relatively quickly. When it gets severe, rehydration therapy is the most important thing to keep you from getting dehydrated from the vomiting and diarrhea. Antibiotics typically are not given except in extreme cases because this would kill a lot of the gut microbes, and those are competing for space and those are helping us fight the pathogens. So we wouldn't want to kill those off because it would just make more room for the ones making us sick to grow and colonize. Okay, so let's talk about that scenario where the disease is caused by a toxin. This is called food intoxication. The term food poisoning is used a lot, and it's sometimes applied to kind of any foodborne illness, but it's really not correct because you're not getting poisoned by the bacteria. It's usually the bacteria are growing and causing a problem. So in the case where it's an actual toxin, that could be called food poisoning. But more correctly, it's called food intoxication. So that's when the sickness is caused by the toxin in our system, whether or not the microbe is still there. The one that causes this is Staphylococcus aureus. They produce something called an enterotoxin or a gut toxin. It's a toxin that harms our intestines under the right conditions. Now, these enterotoxins, they are an exotoxin, so they're protein-based, but these proteins are pH stable, so they get through our stomach. And they're also heat stable. They're difficult to get rid of when you're reheating or cooking food. They're not destroyed by boiling. So something where you warm it up in a microwave or simmer it on the stove to reheat it, that's not hot enough. So that even if the microbes are dead, the toxin is still there. So let's talk about the right conditions where this happens. There are 3 conditions where Staphylococcus aureus will produce these toxins, and all 3 of these have to be in place. First, foods that are salty or foods that have been previously cooked and then handled with bare hands or with unwashed hands. So you've eliminated the competing microbes and you've provided a good environment for Staphylococcus to grow. Remember, salts suppress most things except Staphylococcus. That's why Staphylococcus grow on mannitol salt agar. So you've provided space and a good environment for Staphylococcus. Second, food has to have a good protein source for them to be able to make those toxins. So things like dairy products, things like cheeses, meats, eggs, something like an egg salad or a chicken salad or lunch meat, ham, those are all the kinds of sources that S. aureus would grow on. And third, food is left out at warm temperatures, like out at room temperature for too long. So the bacteria grows rapidly, has this good protein source, has a lot of space, makes this toxin. And then if somebody eats the food, they're getting a big dose of that toxin all at one time. Next, let's spotlight these 3 diseases, shigellosis, salmonellosis, and cholera. First, shigellosis is caused by Shigella bacteria. There's a few different species. They're rod-shaped and gram-negative. You can see them down here in the picture. These are all the bacteria in the background here. These do something kind of cool. They're non-motile on their own. They do not have flagella. But once they get inside our cells, they actually steal our own proteins and use our cells' proteins to build temporary flagella to help them swim around inside the host. They force the intestinal epithelial cells, so that lining, to take them inside. They cause those cells and the M cells to burst, so they're inhibiting our immune system's ability to kill them. They can survive phagocytosis. They can live and even multiply inside phagocytes, and they produce a toxin called the Shiga toxin that damages blood vessels. So this is where we see that bloody diarrhea. This is a stool sample, and you can see white blood cells and red blood cells that have leaked out of damaged blood vessels. So that's something distinctive you would see with shigellosis. Second, salmonella causes salmonellosis. This is usually caused by Salmonella enterica, sometimes Salmonella bongari. This is pretty rare, but it happens. These are also rod-shaped and gram-negative, but these have their own flagella, so they can swim around. At low levels, they're part of our normal microbiome. When it causes disease is when we get an extra dose from contaminated foods. Usually it's eggs or poultry. That's normally where it's carried. They do not survive high temperatures, so proper handling and cooking really quickly and easily gets rid of Salmonella. So where this comes in, problems is when meat is undercooked, or when eggs are undercooked, or when people are handling them with their hands and then get some in their mouth. They also force intestinal epithelial cells to take them in by causing these folds, and then they just kind of tuck inside the folds and the cells take them in. They can also multiply inside phagocytes, and they trigger inflammation in the intestines and hypersecretion of fluids. So our intestinal cells basically just start leaking their cytoplasm. Fluids just start running out, and that's what makes the diarrhea here. Third one, cholera. This is caused by a species called Vibrio cholerae. They're also gram-negative. They also have flagella, but they have this vibrio, this kind of curved shape to them. So that's a pretty distinctive. This is not a very common shape of bacteria. Cholera is pretty easily killed by stomach acid. So you need a large dose to have enough surviving cells to reach intestines. In environments where you have cholera, usually they grow in pretty large numbers. So when you get a dose, it's usually a large dose. They can also attach to those epithelial cells. They make an exotoxin. They makes our cells release ions. Those are electrolytes, things like potassium, magnesium, sodium, and fluid. So again, the cells just start opening up and leaking. This, however, is much more rapid than the others. So this causes massive fluid and electrolyte loss. When I'm talking massive fluids, 12 to 20 liters per day. We only have about 5 liters of blood. So it's difficult to take in enough fluids to keep, like by drinking, this would be like 5 gallons, 4 or 5 gallons of fluid that you'd have to drink to replenish this. So with cholera, the danger is rapid dehydration. For this, you usually need like IV fluids. And these are spread in untreated water. So any place that has sewage or drinking water treatment or heating, if people heat up their water, all of those things remove and kill cholera. But areas that don't have that, here's a map showing cholera, a lot in Central America, Africa, widespread in Asia. Areas with poor sanitation where not everybody has access to sewage treatment and drinking water treatment. This is where cholera spreads. This also spreads during natural disasters. So let's talk about how climate change can affect the spread of a disease. Whenever those sanitation and sewage disposal systems are compromised, you have the potential for major outbreaks of cholera in the untreated water. In Haiti a few years ago, after their earthquake, it damaged septic systems and pipes and sanitation plants. So there was a big outbreak of cholera. So earthquakes can cause those. Other types of natural disasters, things like hurricanes, tornadoes, severe thunderstorms, flooding, those can cause water treatment systems to overflow and contaminate the water. Those can damage pipes and water treatment plants. So all of those things can lead to cholera outbreaks. What does this have to do with climate change? Climate change makes these events more frequent, more severe. They may be occurring in more places or new places. And this increases the cholera outbreaks. Last one I wanted to mention, since this is a pre-nursing class, Clostridium difficile causes something called colitis, inflammation of the colon. That's the end of the digestive system. This is a gram-positive rod, produces exotoxins. It's part of our normal gut microbiome, but after antibiotics, others get killed off, and this one starts to grow like crazy, so it's often a healthcare-associated infection.